So good morning, everyone. I'm very happy to be with you today to present you my research activities dedicated to the wonderful marine worms. And today I will talk about both spaghetti worms, on the left side, and bloodworms. So very exciting. I work in Arcachon Bay, which is a large microtidal lagoon in the southwest of France. Here you can see a photo at low tide, so yeah, a big lagoon. In this lagoon, you can find the largest Zostera nultimido in Europe, which is about 50 square kilometers. This place, Arcachon Bay, welcome many tourists every year, especially during summertime. Here you can see the two famous huts which were used to survey oyster farm during Napoleon period. And here our beautiful Dune du Pilat, which is the highest sand dune in Europe. From the dune, you can see both the ocean and the pine forest. Unfortunately, there was a huge big fire last month. And as you can see on this photo, most of the forest has burned. I mean, it's a small one in comparison of Australian bushfires, but at a French scale, it was a very, very large one. Arcachon Bay is also a major place of oyster farming in France, and it's very important in my work. We will see it later. What you have to know is that after an important disease in the 70s, the European oysters was replaced by the Japanese oysters. And due to this transfer from Japan to Arcachon, Arcachon Bay has become a hotspot of non-native species, with about 100 species recorded in the bay. I mean, 100 exotic species recorded in the bay, and most of them originated from Asia. However, this ecosystem is also well of marine biodiversity, from invertebrates to birds. For my work, I work in EPOC lab, which means continental and oceanic environment and paleo environment. This lab is under the administrative supervision of both Bordeaux University and CNRS. And the main section of our lab concern sedimentology, chemistry, toxicology, but of course marine biology. And in our lab, we have two sites, one site on the Bordeaux campus and one in Arcachon. And I work in the Arcachon Marine Station, which is one of the oldest marine in France, which is very close to the beach. So we have a very nice situation. So my job, actually, I am a research scientist, which is probably the best job in a lab, at least in France. I mean, it's like a technical researcher spending a lot of time in the laboratory. In my lab, I am the manager of the biodiversity platform, and I work in priority on research programs, but also with different company agencies with contracts. And these contracts allow me to finance my research activities. Even with the molecular lab, especially with Cerise, who is the technical manager of this molecular lab in Arcachon. And maybe you have already met her on the second floor at AMRI, as she's working for the moment with Lena in the DNA lab. Finally, as said Pat, I'm getting to know AMRI very well, as it's my third time here. Actually, I have received 3 grants to come to work with the marine invertebrates team. The first one was given by the French Embassy and the Campus France in 2018, and the two other one by the excellent initiative of Bordeaux University for 7 months and 2 months this time. So let's talk about spaghetti worms. The first question is, why studying worms? So I started to work on worms taxonomy 6 years ago. And why I decided to study very, very beautiful. with a lot of different shapes and colors, but also because they're very important for marine environments. They are present from estuary to abyss, from polar to tropical regions. They are also one of the main components of benthic habitat, both in terms of abundance and number of species. And they are also very important for the trophic web, both for benthic fishes and waders. And finally, and more important, there are still many species to describe. And as there are no more French experts since the 80s, maybe, I decided to occupy this vacant ecological niche. So our made path. Each year in France, the French Marine Lab Networks, RESOMAR, organize a one-week taxonomic workshop. During this workshop, we invite an international expert to give us a course on a particular group. And during 1 week, we have practical courses. The expert shares literature, keys, and check our specimen. So in 2016, I invited Pat to give us this workshop in France, which was dedicated to terebellids, like spaghetti worms. And rapidly, we realize, we can see Pat here and me, we realize that we have a lot of problem with French and European species. So, oops, excuse me. So after several discussions over glasses of French wine, we decided that it would be nice for me to do a taxonomic PhD aimed at revising the French species of terebellids. So I offered Pat to be my supervisor, and guess what? She said yes. Rapidly, we were both involved in a European study focused on this beautiful cosmopolitan species, Terebellides troemai, described in Norway by Sars in 1835. So with all European colleagues, we worked on the DNA sequence of hundreds of specimens belonging to this cosmopolitan species. You can see on this map that this species is regarded worldwide. And finally, we realized that we not did just with a single species, but 27 different ones, most of them new for science. So this study confirmed the high number of cryptic or pseudocryptic species in terebellids. So let's go for this thesis. The title of my thesis was Revision of Spaghetti Worms from the French coast, and it was supervised by Pat and by Xavier de Montaudoin, who is professor at Bordeaux University. Few words on ecology of these wonderful worms. So these spaghetti worms are tuberculous, so they have a reduced mobility. They can be solitary or live in dense aggregation as these sand mason worms, which is considered as an ecosystem engineer. They are selective deposit feeders, and they use their tentacle lined with cilia to carry particles from sediment to the mouth. With this video, you can easily understand why we call them spaghetti worms. Oh yeah, we can see the particles moving from the sediment to the mouth. All the spaghetti. So during my thesis, I focused on the 5 different families composing the terebellids, both with morphological observation and molecular analysis. And with the first paper devoted to Trichobranchidae, we described 9 new species from France. And for example, one of them was named in honor of the CNRS that celebrates in 2019 80 years since its foundation. Second one was named after my daughter Lila. With the second paper, we described 8 new species belonging to Polychaetes. One of them was dedicated to Geoffrey, who is editor of Worms website. Does a huge work for the community. And the second one to my friend Chris, who is also from northern from Darwin Museum, and who is also a specialist of this family. The third paper was dedicated to Telepodidae and Telotelepodidae. We described 3 new species, and we recorded the presence of an exotic species for the first time in Europe, Telepus japonicus, and guess what? Recorded in Arcachon Bay among oysters, shell. One species was named after Sue Lindsay from Macquarie University, who is probably the best SCM technician I have ever met. Finally, in the last paper, we describe 11 new species among terebellidae. And in this last paper, I decided to choose some fun species name. For example, one species was dedicated to Kelly Slater, surf world champion, who is also an environmental activist for the project. And as you can see, I have received a short reply from him, and apparently he likes it. So the second one was dedicated to Banksy, the brilliant street artist, whose graphic work always convey powerful messages. And once again, he answered me. His team sent me a message. So he liked it too, but unfortunately, I haven't received any painting. So the reason why I choose to give fun names was also to interest the press and to talk about the importance of taxonomy and studying biodiversity. And I have several articles in the press, but also some interview on the radio, even in my favorite journal, Surfer Journal. So the second part of my talk is devoted to bloodworms. With Pat, we have worked for several years on these worms. They are used worldwide as live baits for recreational fishing. And for example, in Arcachon Bay, they represent an important economic activity as they are collected by 12 companies and employ 26 people. These worms are shipped live by air, by plane, every day in boxes with plant litter to the fishing shops in France. And we estimate that around 1 million of these worms could be collected each year in the bay. Sorry. But due to this important resource, several conflicts occurred in the past among the professional fishermen, and we are now trying to manage the resource. I mean, the price per kilo is higher than lobster, so of course there's some problem. And you can see on this press article the size of the worms, which are very large. So for decades, the species occurring in Arcachon Bay was confused with another cosmopolitan species, Marphisa sanguinea. And following Pat's advice, I decided to do some molecular analysis on several specimens, and the result was very clear. The specimen from Arcachon were different from the Marphisa sanguinea. So we did some SCM, and we found several taxonomic characters to distinguish it from the sanguinea species, particularly with regards to the shape of posterior kitty. So we described it as a new species, and it was named after my son. And for the little story, he's passionate by fishing and is very, very proud to use his worm to fish. But the story was not over. Last time when I was in Sydney in 2020, we did some molecular works on the museum collection. And guess what? DNA of Arcachon species match with specimen from China and Japan. This is Asiatic specimen belonging to another species described the year after Marphisa victory. So we conclude that our recently described species was actually the same as the species occurring in Japan. So we could synonymize it with our new species. And of course, we suggest that the Japanese worms was introduced in Arcachon Bay with juvenile oyster in the 70s. It's not surprising as our specimens are always found in oyster reefs. And as this species, I mean, the Arcachon one is exported live for recreational fishing, we strongly suspect it's present in other localities in France, but also in Europe. So now to finish with also working on species from Pacific, and now we are describing 3 species from Papua New Guinea. These new species are very interesting as they are probably the first one collected from deep sea. And what is funny is that they were all associated with sunken wood, as you can see on this photo. Recently, we had also described a species from India, and we are now describing a species from Victoria, Australia, and another one by Chris from Philippines. So as you can see, it's a never-ending story, and suddenly we have skimmed the diversity of Australian ones. So to conclude, our cooperation is very fruitful, Pat and me, as I obtained 3 grants to come in AMRI. I did a PhD. We have participated to 3 international conferences. We have published several blogs and press articles, but also participated to TV and radio broadcasts involving 44 different co-authors from 11 different countries. So we have still too much to do, and to be honest, it's very exciting. Our cooperation, it's why we have species remain to be described, even in well-known geographical areas as in Australia and France, but because, as you saw before, because these species were confused with so-called cosmopolitan species for decades or more. And our cooperation highlights the importance of mentorship. I have to say that I'm very, very lucky to have crossed paths with Pat. So thank you, everyone. Many thanks to the marine invertebrate teams. It was a pleasure to work with you during this last month. Thank you to the Australian staff to welcome us, Cerise and me, and Megan for the organization of this seminar. Thanks to Sue Lindsay from Macquarie University for the SEM. And thanks, Pat, the best mentor and the queen of Pavlova. So thank you very much. And if